Minnesota, good part, not Heights.
I like that.
What?
Then you said it very much.
So, Jamie has experience here in chemistry and biochemistry and physics and French. He occasionally runs from one point to another. And he's done research, much like Anna, in a very wide span of work. Atmospheric chemistry down at the University of Miami. More in the I'm blanking now. Oh, he worked at Pacific Northwest National Labs doing materials chemistry. And this past summer, he was at Mount Sinai, where he's going to talk about today, doing a little bit more classical biochemistry. Let's see what's open.
All right, thank you. And just again, my name is Jamie Brisbois, and I'm going to be talking to you today about the research I did last summer at the Icahn School of Medicine at Mount Sinai. We'll just keep adding on names as people are earning more money, I guess. So the name of my talk, the research project that I did last summer as part of the Simon group in the Department of Microbiology, was immune response to human endogenous retrovirus, HERV for short, envelope proteins in HIV patients. So we're going to go down and kind of break this down bit by bit. So first of all, just in case you're curious, this is in the white circle is where this Icahn School of Medicine is located. I actually work in this all-black building. And the nice thing about working in a skyscraper in New York is that if your lab is lucky and you face a nice direction, this was actually the view out of my lab bench looking over Midtown Manhattan. So it's a very nice study group in my eyes. So anyway.
And the top.
I wanted to kind of talk about some of the basics of HIV AIDS, just so that you can have that in mind as I'm going through my talk. So first of all, many of you know that HIV stands for Human Immunodeficiency Virus. And this HIV virus, it infects and destroys what are called CD4 T helper cells. And these are immune cells that are required for immune system signaling. So for recognition and then subsequent destroyal of toxic cells, viruses, stuff like that. It's typically thought of as being a sexually transmitted infection, but you can also acquire it through other means, such as from a mother to her child through pregnancy, breastfeeding. If you are a healthcare worker, you have a higher risk just from occupational exposure to blood and other bodily fluids, or if you get a blood transfusion from an HIV-infected patient, you can also are at very high risk of contracting the disease. AIDS is just the late-stage form of HIV, acquired immunodeficiency virus, and this is when so many of these CD4 immune cells have been depleted and infected by the HIV virus that your immune system is basically completely crippled and it can't fight normal infections. And so this is where this leaves you vulnerable to what are called opportunistic infections, meaning the common cold, pneumonia, a viral infection that normally your body could clear relatively easily can prove ultimately lethal because you just don't have the immune system left to fight the disease. So it's really not the HIV AIDS that is deadly, but these other opportunistic infections. The virus is now believed to have originated in African chimpanzees as what is called the simian immunodeficiency virus, which infects and has some of the similar effects in chimps. It is believed that this transfer occurred during a hunter that was butchering an infected chimp, may have gotten some of the infected blood into a cut or wound, and this may have actually occurred on several separate events, which is maybe why we have some very distinct versions of HIV. Just if you are interested, there's this great book by Jacques Lacan entitled The Origin of AIDS that just kind of talks about how this virus may have developed, and the start of the epidemic was really interesting. There are now some treatment options available, but they're not curative. They only treat the disease. There are what are known as Antiretroviral Therapy drugs, and they work just by limiting the HIV virus replication, so delaying the transition of the HIV to this AIDS, this crippled immune system state. But there are still a lot of undesirable side effects, and like I said, they're not curative. There is currently no vaccine because the way the virus replicates, it evolves very rapidly, and also it's a very short life cycle. And so, as of yet, there is no vaccine currently to the HIV virus, as there is to some other viruses like influenza, where even though the virus is evolving, you can make a new vaccine about every year, every 6 months. So I wanted to go over what this HIV virus structure looks like, because what my research is going to focus on. It's very important to part of the structure. And so the HIV virus is very small, about 100 nanometers in diameter, and we can kind of divide the components into three main categories: the structural elements, the binding proteins, and the enzymes, in addition to 2 copies of the viral RNA, because in a retrovirus, the HIV stores its genetic information in RNA, not DNA. So the structural elements, this is a lipid bilayer membrane-bound particle, roughly spherical in shape, lipid bilayer, just like human cells. And this surrounds what is known as the capsid, which is where the enzymes and the 2 copies of the viral RNA are located. And there are also some additional, what are just called matrix proteins, that just help give the virus its shape and structure. Embedded all around in this lipid bilayer membrane are these envelope binding proteins. It's a complex of 2 subunits, GP120 and GP41, and these are what are ultimately necessary for the infection of the CD4 cells. I'll show the mechanism for that in the next slide. And this is also what the human is going to be producing antibodies to, because they are what are expressed to the environment. So that's important to keep these in mind, because they'll come up again. And then I'm going to name the enzymes that are important. There are 3 viral enzymes: reverse transcriptase, integrase, and protease. I'll show where those occur in the life cycle on the next slide. I just wanted to quickly draw attention to those. So in this HIV life cycle, we start with, at number 1, this mature viral particle. And this is just another illustration showing the same thing that we saw in the previous slide. These binding proteins, envelope binding proteins, this complex of the GP41 and GP120, are what can recognize the CD4 receptors, which are scattered all across the surface of these CD4 T cells. And they're what the cell can use for this immune signaling. But the HIV virus can bind to these, and when this binding event occurs, causes a structural change in the conformation of the proteins, such that the lipid bilayer membrane of this viral particle becomes absorbed into the lipid bilayer membrane of the CD4 cell. And this is where, at number 2, this fusion event occurs. When this occurs, the contents of the viral particle that were in this capsid, the viral RNA and the viral enzymes, are transported into the cell, where at number 3, this is where this viral enzyme, reverse transcriptase, can transcribe the 2 copies of the viral RNA into DNA. That is then, at number 4, shuttled into the nucleus through one of the nuclear pores on the nuclear membrane, where, again, a viral enzyme, integrase, will make a cut in the viral DNA and insert make a cut in the host DNA and insert the viral DNA. Then, at number 6, the host's cell machinery will take over and produce many copies of these essential viral proteins and enzymes in the viral RNA, which, when exported again out of the nucleus, can butt off at number 7 into a new viral particle. Once this occurs, we get a little bit of reorganization at number 8 to get back to number 1, and then this viral particle will proceed to go back to further stuff. So what are these endogenous retroviruses? Well, they are like HIV in that they are retrovirus, that their genetic information is stored in RNA, but these are actually remnants of a past ancient infection of a retrovirus several million years ago in human history. And so now these retroviruses have become part of our germline, meaning that they are completely integrated into our genome, and that's why they're called endogenous. In fact, at least 8% of the human genome can be traced to retroviral origin, which was kind of shocking to people because that's actually a greater percentage than gene coding regions. But it's not as scary as it sounds, because most of them are actually inactivated, meaning there are various ways the cell has to combat this type of insertion. Insertion, deletion, and what's known as heterochromatinization, which just means that the viral genome is not going to be in a state that it can be actively expressed. Of these HERVs, a subtype, HERV-K, are the youngest known integrated retroviruses, and why those are of interest is because they are most recently acquired. We would expect that they would have the least amount of this inactivation, so that they're going to still retain some either partial coding ability, meaning some viral proteins can still be made in the right circumstances, or that they may be able to try and produce a viral particle that would be similar and analogous in structure to the HIV virus that I showed on the previous slide. expression of this HERV-K subfamily has been associated with immunocompromised states, such as various cancers, Hodgkin's lymphoma, breast cancer, testicular cancer, to name a few, but also what was going to be important to my research project, viral infections, Epstein-Barr, and HIV-1. So how do these HERVs relate to HIV? Well, it has previously been shown that these HIV patients mount a specific immune response to these HERV-K envelope proteins, meaning that they will have both T cells and antibodies produced responding. And also that in these immunocompromised states, these otherwise inactive HERV elements can start to produce virus-like particles, meaning structures that look kind of similar to the HIV virus with these envelope glycoproteins expressed on the surface. And that these seem to be expressed at higher levels in HIV patients and some of these cancer patients. And so the rationale that led to my research project was that an antibody response that would be targeted at these HERV envelope elements expressed in exclusively HIV-1 infected cells could then be used as a biomarker to know which cells are HIV infected. As a therapeutic aspect, why this is of interest is because these HERV envelopes being integrated into our genome are not mutating very much or at all, especially compared to the rapidly evolving HIV. So this could be a handle on the HIV virus that would not require changing the vaccine or some of the therapy as much because the HERV elements are much more stable. So the goals of my research project were to first optimize what's called cell-based ELISA. I'm going to describe that on the next slide. To detect antibodies to these HERV-K envelopes in blood plasma samples, so blood samples from HIV-infected patients, and also some negative controls. And then, if I succeeded in optimizing that, I wanted to move on and see how many of these HIV patients are going to have this antibody immune response to HIV first off, and one or more of these HERV-K envelope glycoproteins. And then to test different HERV-K envelopes that we had in the database to determine how specific this antibody response is. If the HIV-infected patients only respond to one, or if they respond to just across the board all sorts of different HERV-K envelopes. So in doing the cell-based ELISA, even before you can get to the actual immunoassay component, you have to prepare your plate with a cell line. So the first thing you do is you just have to coat your 96-well plate with polylysine, which is a polymer of lysine. It's an amino acid that's positively charged. And this enables the cells that we are going to plate to adhere well to the plate, to the bottom of the plastic plate surface. This is necessary because the assay, the ELISA assay, involves a lot of wash steps, and if you don't do this polylysine treatment, they can wash off, and then you're going to have to redo your assay. So you just give about an hour incubation of this polylysine, then you can plate your cells. These are adherent cells, like I said, meaning that they were going to adhere to the bottom of the plate. The cell line I ultimately chose after testing several were 3T3 cells. These are derived from Swiss mouse embryo cells, and the reasons I chose these were, first of all, they were very adherent. No matter how many times we did these wash steps, they seemed to stay stuck to the plate, so that was a good criteria. Second, what I'm going to talk about next, they transfected really well, meaning that we could get them to express these envelope glycoproteins we were looking for antibodies to at a relatively high efficiency. And third, because we're doing an immunoassay, there's going to be some amount of background immune signal, and we want there to not be too much of this background immune signal. And these 3T3 cells just did not have as high of background immune signal, so that's why I ultimately chose them. So you plate these into each of the wells on your 96-well plate. You give them 24 hours to get reacclimated to the environment to grow and divide, and then you transfect the cells. So what you do is you take a plasmid, which is a circular piece of DNA that has a region that encodes for either the HIV or HERV-K envelope glycoprotein. This is represented by the blue. This would be a region part of this plasmid. And what you can do is if you incubate that on the plate with these cell lines, they'll actually uptake the plasmid, this piece of DNA, and start to produce many copies of either this HIV or HERV-K envelope proteins. And just as a reminder, these are those proteins that you would see either on the surface of the viral particle or of on an infected CD4 cell. So we give them 48 hours to uptake this plasmid and to produce copies of this HIV or HERV envelope protein, and then we can proceed with the actual ELISA, where we incubate with the patient samples and look for immune response. Then you have to develop the results, and I tried two different methods. First was a colorimetric method, where the cells are stained red if there's a positive immune response. And then I also tried a luminescent method, where if there is an immune response, the cells will produce light. And I'll describe how that works in a minute. And this is, again, this reminder. So we're looking for antibodies, meaning an immune response to these either HIV or HERV envelope proteins, meaning these green, represented by these green structures that are scattered on the surface of this either HIV viral particle or this HERV virus-like particle, or on this surface of an infected cell. And so ELISA stands for enzyme-linked immunosorbent assay. And I'll say why the enzyme part is important in a minute. And so what this means is that we are looking for antibodies in the patient blood to our antigen. In this case, our antigen is either the HIV or the HERV envelope glycoprotein, represented by the red dot. And so now that we have transfected these cells, meaning they're going to be displaying many copies of the envelope glycoprotein on their cell surface, we have what is represented by the yellow cells expressing many copies of the antigen, these HIV or HERV glycoproteins on their surface. If we do some initial washing and blocking steps, we can then proceed to incubating with the primary antibody. In this case, the primary antibody is what comes from the patient blood samples. So what you would do is you would incubate with the dilution of the patient blood samples, and if there was an immune response, meaning if the patient had an antibody represented by this T, this antibody would then go and bind with a strong binding to these antigens, these HIV or HERV envelope glycoproteins. We then have to do additional wash steps to make sure that we don't get any non-specific immune response binding, and then we proceed to a secondary. The secondary antibody is just going to recognize any human antibodies present in the system. It's a broadly neutralizing antibody. And so wherever there is a primary antibody found in the blood in the first step, we will get an additional antibody represented by the green that will bind and recognize that. This antibody has been genetically fused to an enzyme, which is going to be important in the next step, because we can then incubate with the substrate, either one that will induce this color change or cause the production of light, and then we use that change, either the production of light or the change in color, to give a correlation to the amount of immune response present. And so when we image the results after ELISA, this would be an example of what cells would look like we developed using this colorimetric method. So wherever there is a positive immune response, the cells will be stained red, because the enzyme linked to the secondary antibody is going to cause a chemical change in the substrate, and then the cells will turn red. If there is no immune response present, there will be no enzyme to cause this chemical change, and the cells will not be stained. In the luminescent method, this is where you incubate with luminol. So wherever there is this secondary antibody bind with the primary, with the enzyme, it'll cause a reaction so that the luminol will produce light. So up here, this would be an example of a well with a positive immune response where you get light production. A little bit lower, where there is sunlight production, but it is not quite as apparent, this would be reminiscent of an immune response, but not quite as strong as in these wells. And down here, this would be what would be known as a negative immune response. And so if we look a little bit at how this colorimetric assay would work, I was able to use it to detect HIV antibodies to the HIV envelope, which we would expect, since these patients, being HIV positive, are going to produce antibodies to these HIV elements, and possibly a weak antibody response to these endogenous retrovirus envelopes, these HERVs. The problem with this assay is that because it's colorimetric, and while technically you should be able to put it in a plate reader and be able to measure the amount of red that way, it wasn't working very well, and so it was only qualitative, meaning that we could only collect the data by looking at it. Is this well read or not? And so that becomes challenging when you're looking at very low levels of immune response. But just if we go through, these are some pictures taken of representative of wells in one of the ELISA assays that I ran. First off, I just want to point out that because we were infecting with a plasmid, we need to make sure that the patient samples aren't just producing an antibody response to the plasmid, since that is a foreign piece of DNA. So we use an empty plasmid, meaning a plasmid that does not contain the gene to produce either one of these envelope glycoproteins. And we look to make sure that compared to the top two, these are both HIV positive patients, the bottom is HIV negative, healthy control. There is no distinct red cells in any of the wells. So we can say, okay, we are confident that the patients are not just producing an antibody response to the plasmid itself. Next, because we are trying to distinguish an immune response between HIV positive patients and HIV negative, we want to make sure that the assay would work to do this distinction. So this HIV envelope JRFL is just a commonly used example of one of these HIV envelope glycoproteins. These proteins, binding proteins, are expressed on the surface of the viral particle, and as we would expect, the top two, the HIV positive patients, have a very marked immune response. There are a lot of red cells compared to the negative control. And so this is evident that this assay can at least distinguish between an HIV positive and an HIV negative patient. We already have assays to do that, but at least shows that this assay at its base is working fundamentally. So then, if we go and move over to these HERV elements, which are going to be similar, they're binding proteins like HIV, but they're the ones that are endogenous, meaning that they're already contained in our genome. It's possible that there is some slight immune response in the top two, the HIV positive compared to the negative, but it's very easy to tell that it's very hard to distinguish between if there are perhaps some slightly red cells or not. And this is where this colorimetric assay method starts to kind of fall apart. And so perhaps there is some weak signal in the top two to what is known as the HERV-K consensus, meaning just kind of an average of several different examples of this HERV-K envelope in the environment, but it's hard to tell. And similarly, in the HERV-K N2, which is just one example of another one of these endogenous virus envelope binding proteins, perhaps there is a weak immune response to the top two, the HIV positive compared to the negative control, but it could be just as much wishful thinking after doing this for 8 weeks. And so you need to have a quantitative method to actually compare these. And so that is where, right near the end of my internship there, unfortunately, we switched over to developing the assay using luminescence, meaning that, as a reminder, now wherever we have an immune response, we can measure this using the production of light instead of the production of color. And the nice thing about this is it's very easy to take your plate, put it into a plate reader, and get what are known as arbitrary light units, which means the amount of light produced, and use that as a correlation to immune response. So if we look here, this is an example of the immune response to this HIV JRFL. So this is HIV, not the HERV. These are just 9 HIV positive patients, and their varying immune response measured by, again, arbitrary light units, which just means the amount of light present and thus the amount of immune response. What's interesting to note here is it confirms that our assay is working and that all 9 of the HIV patients have an immune response to this HIV envelope protein, but also that this reminds us that there is lots of variation in the population, both in the HIV-infected population, since these patients all have different immune systems, they're going to have a slightly different immune response even to the same HIV component, but also that, as you remember that this HIV virus is rapidly evolving, that these patients, even though they are HIV infected, what HIV they're infected with is going to be slightly different. And so, for example, if we look at just what's HIV number 1, the start flu, this patient has a much higher immune response to this JRFLN than to what's HIV number 7, the light flu. And this could be attributed to a lot of things, but one example would be that whatever the antibody the HIV1 patient has is more similar to the HIV that patient number 1 is infected with, and HIV7 is just infected with a slightly different version. That would be one way to analyze these. Another interesting thing that I looked at using this luminescence method, again, this is only with the HIV N. I didn't really have time to properly do it with the HERV-Ks like I would like to initially, but this shows us that we have an immune response to multiple HIV envelopes. There are actually different subtypes of this HIV-1. There are 4 that are known, O, M, N, and P. M stands for main. Over 90%, I believe, of patients are infected with this subtype. Of the O, there are only about 2% known that are believed to be infected. And then N, I think they have 10 cases and then the P, they have like 1. So the more likely than not, all of these patients, these 9 HIV patients that I was looking at, were most likely infected with the M subtype. That said, as we would expect, this is on the bottom, we have a GP160MN, which is the precursor to this envelope binding protein that's expressed on the HIV surface. But for that main subtype, and the other two, the O and N. As we would expect, since these patients are most likely infected with the M subtype, almost all of them have a larger immune response to the GP160MN envelope. But what's interesting is that even though they are most likely, like I said, infected with the M, they also seem to have somewhat of an immune response to the 2 other subtypes, O and N, I tested. This suggests multiple things. Again, this could just be attributed to variation in the immune systems of the patient, and also to the variation in the viruses they're infected with, but also suggests that there's probably some homology in the structure of these envelope proteins, even though they come from different subtypes, such that the antibodies that the patients are producing to the GP160N are at least somewhat effective in binding to the envelopes of the other two, the GP160O and the GP160N. And just the last point, granted this is the only time I ran this assay, so the levels have to be taken with a grain of salt because they're not done in triplicate or anything. But even so, if we compare the proportions of the immune response among patients, for example, if we look at the red HIV positive number 4 as by far the greatest immune response to the GP160M. But if we look at the GP160N, one of these other subtypes, it's actually this HIV number 2 that has a greater immune response. So again, this is showing us that there is varying magnitude and selectivity of the antibodies produced by all of these HIV-infected patients to different variations of the HIV and the subtypes. So just to summarize my results, I did succeed in doing some initial optimization of this cell-based ELISA assay, this enzyme-linked immunosorbent assay, to look for antibodies in patient blood samples to both the HIV and the HERV envelopes. And of all the 8 HIV positive patients that I looked at, they all did have an immune response to the HIV envelope using the assay, which again, there are already assays that do this, but it does at least show you that your assay is undergone an initial optimization step. Third, that, like I just showed in the previous slide, these HIV patients seem to have an immune response to multiple subtypes of HIV, even though they're only infected with one, most likely the M subtype, the main subtype. And last, that perhaps when I showed you those pictures of the red cells, perhaps these HIV positive patients are exhibiting an weak immune response to these HERV envelope components. But that was hard to tell using the colorimetric method, which is why moving forward, we would want to continue optimizing the assay, now using the luminescent model for the detection of this immune response to these HERV envelope proteins. And if we could get the assay to a relatively high level of efficiency and fitness, then we could just proceed to test all the patient blood samples that we have in our bank against all of the different HIV and HERV envelope binding proteins, and then start to draw a conclusion about how frequent and selective the amount of antibodies this immune response is. And then long term, like I mentioned near the beginning, that if we can start to correlate and find a correlation between antibodies to HERV envelopes and HIV-1 infection, perhaps this could be a therapeutic target that is more stable than that of the rapidly evolving HIV virus. And so that's why there's a lot of research going on in this area at the time. And so I would just like to acknowledge everyone in the Simon Laboratory. Dr. Viviana Simon was the head of the group. I worked primarily with Ben Mulder, and then I want to acknowledge also other people that were there for inviting me and helping me in my internship this summer through the Mount Sinai School of Medicine CERT program, which provided me the housing and funding to live successfully in Manhattan for a summer while doing science. To the Lawrence University Chemistry Department for allowing me to give you this talk today. And to my chemistry advisor, Stephen Dever, for giving me support throughout the 4 years here at Lawrence. So I'd be willing to take questions at this time.
So this is just to understand the assay better. So you used you were using those mouse cells and you were having those cells express these envelope proteins, and then using the human blood as trying to get at the human blood's antibodies.
Correct.
To test the, okay.
Yeah, so our primary antibody in this case, we're just looking for the presence of them in the blood samples. So we don't know if they're there or not. And that is why when we follow with the secondary, which is just going to recognize broadly the anti-human, then we can develop it using the various methods to see if there are any antibodies present. So it's kind of a prospecting for antibodies method. Yeah.
Discussing the difference between the envelope 18.2 and envelope consensus, was that simply the plasmids had, and the consensus was the main sequence and the 18.2 was a specific variant, or was it a consensus of quantity?
A consensus, there are lots of these different HERV elements that are present in the genome, and there of course is variation among the population. And so the HERV consensus is just kind of like an average, a genetic average of those, and hoping that that would be the most conserved elements. So predicting that most patients would have an immune response to that. And HERV 18.2 was one specific subtype of these HERV envelope elements that previous research has suggested might be upregulated by HIV. And so that's why we're looking at that one. Rachel?
You mentioned that you had 8 patients and they all showed good immune response. Are there people that don't show immune response to HIV?
It's also important to note that these patients were all on antiretroviral therapy, meaning that the amount of antibodies they're producing is probably going to be lower than someone that's initially post-infected. And I didn't mention it, but the amount of HERV present normally increases just after infection, but you can't just find recently infected HIV patients normally to get this kind of data from. Normally you get blood samples from people that are on the long-term treatment. And so there's also the possibility that even though there could be antibodies present, these patients are just too long in their course of the disease and on their therapy that they could just be so low that we could not be able to target that. Yes?
So are there a lot, like, when you're talking about the consensus, so the HERV envelope you're referring to envelope proteins, correct?
Yeah.
So the consensus is just one protein that's commonly. But there's a bunch of different HERV proteins.
Yes, this would just be one that's made in vitro, just through the genetic recombination, designing it to be more or less an average of the commonly known ones, but it would hopefully have the most conserved elements, and thus the antibodies, even though they're made to different HERV elements potentially, will still recognize it to some degree.
So you showed, just flashed up there, that you used a 1 to 5,000 dilution of the sera. How did you decide what that concentration was?
Well,
what that dilution
so that was
should be?
like about half of my internship doing that.
Okay,
And
that's
it
what
turns out
that's
we
what I
should've-
thought.
And it turns out we should have started about 1 to 5,000. We were starting at like 1 to 20, and there was just so much background immune response that you couldn't distinguish. Even the HIV, which just to specify, there are going to be many more antibodies to the HIV than perhaps even to this HERV. And so if we can't even distinguish the immune response to HIV, we're going to have no luck finding the HERV. And so you have to, this is the difficult part, which we didn't succeed in completely optimizing, where you need to dilute it enough where you can get out the background signal, but not dilute it so much where you're not going to be able to find these HERV antibodies if they are there at all.